delighted to welcome our seminar speaker um, all the way from Davis, California, Louie Yang, who's going to be talking to us about the very most topical subject that we could think of right now, which is the periodical cicadas. And if we were meeting in person, you could be sure that besides Gina and I wearing our cicada outfits, and I would like to advertise the earrings that John Lill's daughter is making out of cicada wings, and my cicada shirt, and my coffee mug. We really, John's putting on his mask, and Gina and Austin have their shirts on. We really like to go all out, and sometimes it's more challenging to dress for our speakers, but we do our best. But in any event, if we were here in person, I promised that I would have a little plate of chocolate covered cicadas on the table for people to try. And I expect that there would be almost as many at the end of the seminar as there were at the beginning, because some people draw the line, but we're trying to expand our willingness to eat tree shrimp in different incarnations.
Very nice.
So anyway, Louie, thank you so much for coming to talk to us today. And let me tell you just a little bit about our speaker. Looking at his biography, I was surprised to learn that Louie was born in Australia and grew up in West Virginia, neither of which I knew. He studied for his undergraduate at Cornell. He did his PhD at UC Davis with Rick Carbon. And what did he study for his PhD? Periodical cicadas. He has been on the faculty in entomology and nematology at Davis since 2009. And he's a community ecologist, particularly interested in temporal dynamics of ecology. And as John mentioned at the beginning, he has done some really important fundamental work on phenological mismatches related to climate change. He's very interested in how timing explicitly fits into ecological interactions. Near and dear to Gina's heart because he's been looking at resource pulses and the effect of resource pulses. Near and dear to John and my hearts because we have been looking at differences in host shifts and ability of caterpillars to survive predation during different generations of the year, and looking explicitly at the temporal issues. And so his temporal focus has really been a nice anchor to look at community ecology questions.
And can I just say he's near and dear to my heart because we just put in a proposal together.
Exactly. So, he's pretty much an honorary Georgetown faculty member. And with that, Louie, please tell us about periodical cicadas as resource pulses linking above ground and below ground communities, a 17-year retrospective.
Oh my goodness. Thank you so much for the invitation to come speak here. And it's so nice to see everyone's faces. It's such a strange time, and it's great to be able to see colleagues and friends, even if it's virtual, and to be beamed into living rooms and offices and, kitchen tables in the cicada range, even though I'm not going to probably make it there in real life. It's great to be able to see everyone. It's been a strange year. I feel like last year around this time, the public consciousness was synchronized and focused on toilet paper and other aspects of survival. And it's nice a year later to have, another synchronizing event and have the public consciousness focused on these insects that are emerging from below ground. So I'll take it. It's great to be able to think about these insects that have been there the whole time. I also can tell I like this department already just by the chat window. I hope you keep that chat window coming. I'll try to read the chats as they come up, and if any questions come up, I certainly encourage folks to interrupt or put it in the chat or whatever it takes to get our attention and try and answer those questions.
And
So
I'll
thanks again.
monitor the chat for you as well.
Awesome, thanks.
And I'll send you a link to it at the end so you can read all the great comments. So don't worry about missing anything.
Awesome, awesome. So I'm really glad to be here. I didn't think, I realized that the timing would be so close to the actual cicada emergence. They seem to be coming out a little earlier than I expected this year, but I'm glad to be talking about periodical cicadas. It's been 17 years since my first cicada paper. It's strange to think I've been doing this for about 17 years now. I think I've learned a lot from periodical cicadas over the years. There are three themes that I think I'm going to try to address in this talk. Three things that I think I've learned from periodical cicadas. The first is that resource pulses show the structure of species interactions. Resource pulses are these events like periodical cicadas that really perturb communities, and they allow us to see how species are connected. Second is I think that periodical cicadas illustrate this deep link between two schools of ecology that have been disconnected for too long. This community-based school of ecology focused on organisms and species interactions, and this ecosystem-based perspective of ecology, which is focused on nutrient fluxes and abiotic drivers. And I feel like cicadas allow us to see the links that have always been there between those two schools of ecological thought. And third, I feel like cicadas have made me think more about time, and in that way have really deeply influenced my view of ecology. I've come to this conclusion that we need a more temporally explicit perspective in ecology. And I feel like thinking so much about timing during all those cicada emergences has really influenced the way I think about ecology. So I'm going to try to illustrate those 3 points in the course of this talk. This is also a kind of indulgent personal retrospective for me. So I hope you will humor me. In a lot of ways, the research that I've done with cicadas really has taken up most of my academic career. And so when I look back on the research I've done, it's kind of impossibly nostalgic for me, especially this year. 2004, I was a struggling graduate student driving around in a beat up old car in the mid-Atlantic states, trying to collect data as frantically as I could. I was just starting to study cicadas. I'd been doing it for a couple years, and Brood 10 was the big one. And my first cicada paper was published, pretty much 17 years ago. I was 27 then. And I'll be 44 at the end of this year. And in a lot of ways, it feels like not that much has changed, but in other ways, it feels like everything has changed. And so taking a look back on 17 years, it does make me realize that I've decided that one cicada generation is a good unit for measuring an academic career. I feel like I'm lucky to get 2. If I'm fortunate, I might get 2 cicada generations. I'm not gonna, push it. I feel like that's something we can shoot for. And it sort of made me realize that you only get one shot at a lot of things. You only get one shot, and our time is short. So let's move forward. So I've broken the talk up into parts. The first part is going to focus on this first theme, that resource pulses allow us to see the structure of species interactions. There'll be 3 parts in total. This first part is the longest, and I'll try and stop between the parts to see if there are any questions. So, the other thing I'll say is that it's been fun to go back through some of my old talks and dig up some old slides. It's like finding the box you stored in your garage, and finding these old slides from a long time ago. So this is, this talk has parts of talks that I've given for so long. It's really fun to revisit them. So this is a quote from a review in Trends in Ecology and Evolution from 2000, where Rick Osfeld and Felicia Keesing wrote that many terrestrial and aquatic ecosystems are characterized by pulsed resources. And they suggested a lot of examples of pulsed resources. The massed production of acorns in a large area of North American forests, the mass production of beech in other forests, the die-off of anadromous fish that move marine biomass into terrestrial rivers and streams and then deposit the biomass there. Insect outbreaks of many kinds. These are locust outbreaks in North Africa, but there are many kinds of insect outbreaks that may represent important resource pulses. Weather events like El Nino events in the West, where these large global patterns in sea temperature create wet years and dry years. And the wet years can be a big pulse of a very limiting resource in these arid ecosystems. And then on the east, in the Atlantic, these hurricane events create disturbances, which also create resource pulses, either opening up the canopy in tropical forests or delivering a lot of foliar nutrients to the soil in tropical ecosystems. So these are a few examples of resource pulses, and I've just sort of chosen 5 of them here to illustrate. But I guess the main point that I realized upon thinking about it more is that there are lots of examples. If I hadn't chosen these 5, I could switch it up and choose 5 other examples of resource pulses. Resource pulses really do seem to be widespread in nature and affect a lot of ecosystems. So let me take a step back and define what a resource pulse is. I think a resource pulse is an event of increased resource availability in time that has 3 characteristics. It's relatively infrequent. It's not happening all the time. It's of relatively large magnitude. It's an unusually large input. And it's of relatively short duration. It's not a press, it's a pulse. And so we're talking about these events that are infrequent, large magnitude, and short duration events that perturb ecosystems. They kind of knock the system off balance, and we get to watch it respond. Periodical cicadas are clearly an example of a kind of resource pulse. You all probably know as well as anyone the life history of periodical cicadas, but I'll briefly review. They are below ground root feeding herbivores, and they spend 13 or 17 years, depending on the species, feeding on plant roots below ground. And during this time, they are relatively safe from most predators. So they are growing, developing, increasing their own biomass, and they're doing it all below ground, largely unseen and largely forgotten by the above ground community. And then in this kind of remarkable event of synchronized life history, millions, perhaps billions of them come out of the ground, all basically within the space of a week or two, across a very large area of their range. The upcoming Brew 10, as you probably know, is the largest, and it covers many, many states. It's quite a phenomenon of nature to see. Going back below ground, periodical cicadas go through 5 nymphal instars before emerging. And then when they emerge, they usually emerge at night or in the morning towards dusk, and they will come out of the ground, climb up nearby vegetation or structures, and then the adult will emerge from the nymphal exoskeleton. And when the adult emerges, they are white. They're teneral. Their exoskeleton is still soft, and it takes a few minutes to a few hours to dry out and harden and darken. Then the adults will go through courtship and mating. This is where they're making a lot of noise with courtship chorusing and mating. Then the females will lay eggs by inserting them into twigs. She has a hard ovipositor that she will use to make small incisions into pencil-sized twigs, often in sunlit areas of the forest. These eggs will develop long after the cicadas have disappeared from being so apparent in the system. And around August, the eggs will hatch out and very small nymphs will drop to the ground, burrow through the soil, and try to find host plant roots to attach to and begin feeding. And then they'll start the cycle again. So most of what we see and most of what gets the attention of the public is this adult stage. But cicadas spend most of their time below ground, and we often don't have evidence of it. But when they do start coming out of the ground, we see these emergence holes. And I've heard a few reports from friends and colleagues that these emergence holes are starting to appear as the cicadas work their way up through the soil. So when I was a graduate student, one of my first questions was, how do these cicadas affect forest ecosystems? And it was a pretty broad question. One of the things that's apparent about cicadas is that they are not particularly good at avoiding predators. They've been described as predator foolhardy, meaning that they seem distracted by other things and not particularly concerned about capture. And so they'll land on you. They'll bounce off your house. They'll be easy to catch. And they're easy to catch for ecologists, but also for a variety of predators in the ecosystem. One of the things I most love about cicada emergence events is that predators that don't normally eat insects start to eat them, and they behave erratically as they recognize this new resource. This is one of the things that I think is very interesting about communities, is how ecological communities are able to respond flexibly to changing conditions. And so when a new resource presents itself, some organisms will try to eat them, and if it seems like a rewarding thing to eat, they'll continue eating them. And so here's a cedar waxwing that was eating a cicada, I guess about 17 years ago now. Other predators will eat them, spiders will eat them, a lot of things that don't normally eat insects will eat them, turtles, snakes, fish. I've seen a lot of creatures start eating cicadas during the cicada emergence event. And it's pretty apparent. It's not something you have to look really hard for. You'll start to see it if you're out hiking around during the emergence event, especially in the early days. But one of the things that always struck me as particularly interesting about this phenomenon is that such a small fraction of cicadas are actually consumed by predators. When they emerge at high densities, they effectively satiate their predators. The predators eat as much as they can in the first few weeks, and then they just can't eat anymore. They are, quite literally satiated by this synchronized event. So at reasonably high densities, the proportion that get eaten by predators goes down to a very, very, very small percentage of the total population. This is super interesting because it defies the norm in ecology. It's often the case that nature is red in tooth and claw, and the fate for many organisms is eventually to get eaten, to get sick and die, but often to get eaten before you die. And so cicadas defy that, and that raised a lot of questions for me about what happens to their bodies. These questions they were new to me, and I think most of cicada ecology had focused on the really interesting stuff that happens above ground during the period when they're active and flying around. But I was interested as a graduate student to read some really old literature that had observed the dead part of cicadas as well. And when I say old, I mean really old. This is a paper from 1666. The very first volume of the very oldest scientific journal, the Philosophical Transactions, Volume 1. And this is an article actually by the publisher, Oldenburg himself, who reports upon a second or third-hand account of periodical cicadas in those new American colonies. They describe them as a strange kind of fly. in this really old paper, which you can now download on JSTOR, they describe that when they repose themselves upon the ground, they cover it 3 or 4 inches thick. And if a wheel pass over them, they emit a stench hardly to be endured. And so if any of you were around 17 years ago in a neighborhood that had a lot of cicadas, you might remember the smell of them as they come out at high densities. A lot of them don't make it. A lot of them die. You can smell them, especially when they're at pretty high densities. So 17 years ago, I was in, I think this picture is from the Blandy Experimental Forest in Virginia, which is not too far away from y'all. These are cicada nymphs that came out of the ground the night before, and there was a rainstorm. Often rainstorms seem associated with the high densities of emergence. And these poor souls died in a roadside puddle. After developing diligently for 17 years, they didn't make it to adulthood. They died in a puddle and were just sort of dead carcasses on the road the next morning. If they make it, they develop into adults. They have this teneral phase where they're shockingly white, and then they harden into adults. And many of them will end up like these. They simply die and fall to the ground. This is a site I called Yellowgate, which is somewhere in Maryland. I don't know if, I'm sure I have the GPS coordinates, but it's out on a forest road in the middle of a national forest somewhere. And the road was just conducive to seeing these dead cicada carcasses. As a second or third year graduate student, I used Photoshop to highlight all the carcasses and then remove the background so you could see the density of carcasses littering the road. It was just carcasses as far as the eye could see. So cicadas come out at high densities. The densities can range from noticeable, less than 7 per square meter, to ridiculously high. 350 per square meter is the highest recorded in the literature, but they can reach tremendously high densities. Even if there are more than a few dozen per square meter, it seems like a shockingly high density of very loud insects. They also occur over a very large spatial extent. So they cover a wide range of forested habitats throughout eastern North America. There are several temporally disjunct broods, which is to say that they don't all come out at once across this large range. They come out sort of brood by brood. So all of this suggests to me, or suggested to me as a graduate student, that dead cicadas are likely to be a pulsed resource. They are infrequent, they're of large magnitude, and they're of relatively short duration during this emergent summer, the dead bodies. And so I revised my question to ask, how do dead cicadas affect forest ecosystems? So looking at a cartoon of a community like this, I imagine cicadas taking nutrients from plants for a long time, 13 or 17 years, and then in a pulsed way, delivering it back into soil detritus in the emergent summer, creating new pools of soil detritus that are consumed by detritivores, soil microbes, potentially increasing the availability of soil nutrients in its mineralized plant available form, which then might be taken up by plants as fertilizer, and these enriched plants might affect subsequent other above ground herbivores in the system. And I started doing experiments where I think my very first experiments were a version of this, where I created forest plots. I created plots in the forest, and I used dead cicada carcasses to supplement them to create a patch that was fertilized by dead cicada carcasses. Usually the dead cicada carcasses weren't very difficult to obtain. I measured as many things as I could. I measured the detritivore community and noticed that it makes a pretty obvious shift towards carrion consuming detritivores, things that I wouldn't expect to see crawling around on the forest floor, like carrion beetles and other kinds of carrion-eating insects became very common in these patches. I also noticed that these insects, these detritivores, seem to have a strong effect on the cicada carcasses themselves. So the interaction was reciprocal. The dead cicadas changed the detritivore communities, but the detritivore communities also changed the dead cicadas. If you exclude the detritivores, you can slow the decomposition of these carcasses down by about 40 times. You can make them relatively persistent in the environment, but if you allow these detritivores to access the carcasses, they very efficiently and very effectively shred it up. And that's why these piles of dead cicadas don't persist much past the cicada summer. They are very efficiently mobilized and shredded up into small pieces. Microbial action takes over, and they go right into the soil. Often the only thing you find later are the wings, which are remarkably resistant to decomposition. So I did more experiments, this time focused on the soil microbes, which seemed like they would be the beneficiaries of this shredded up organic detritus. This time using a phospholipid fatty acid analysis. Phospholipid fatty acids are components of cell walls, and they are different between fungal microbes, soil microbes, and bacterial soil microbes. And was able to observe that in cicada supplemented plots, the biomass of bacteria in the soil increased, and the biomass of fungal microbes in the soil also increased. These effects were observed 28 days after the addition of dead cicada carcasses, and seemed to create a trail of interactions that I wanted to follow. So it was a 12% increase in bacterial biomass and a 28% increase in fungal biomass. What does that do to soil nitrogen? So I did another experiment, again supplementing dead cicada carcasses across a range of densities to ask what happens to soil nitrogen. And this time I used ion exchange resin bags. These are small bags of specially doped plastic beads that absorb mineralized nitrogen from the soil in a way that's intended to simulate plant uptake, kind of like plant roots. And then you dig out the bags a few days later and extract them in the lab and figure out how much nitrogen was in the soil. So a priori, I had the expectation that cicada biomass should increase the availability of mineralized nitrogen. So nitrogen in cicada biomass is in organic forms. It's bound up in protein and other parts of cicada bodies. And the microbes are able to break that down into parts that plants can take up. But they don't always make it available to plants. If, in fact, they take up more of that nitrogen into their own biomass, they may actually make it less available to plants. So there's sort of this below-ground competition between microbes and plants. And the degree to which they have excess nitrogen to be made available to plants depends in large part on the percent nitrogen in the biomass that's being amended into the soil. And so a priori, there was a framework for assessing whether we thought there would be net mineralization. So, here we go. So if an input has about 1.5% nitrogen, it should lead to slight immobilization. If, on the other hand, the input has about 2.5% nitrogen, it should lead to slight mineralization, a slight excess of nitrogen in the soil. Cicada biomass is about 10 or 11% nitrogen. And so it's just so much higher in nitrogen than the kind of thing that typically is dumped into the soil, leaves and plant matter, that there's a strong expectation that we should see net mineralization. And in fact, that did happen. These figures are on a log scale, so it's a little bit hard to assess, but the availability of ammonium in the soil increased by more than 300%, and the availability of nitrate in the soil increased by more than 200%. And these effects on nitrate persisted for at least 100 days after the pulse. And so these two pools of mineralized nitrogen seem to increase dramatically in response to higher fertilization rates, and persisted for at least 3 months after the pulse. So this changed my understanding of how this system was working. Up until these, I saw these results, I thought of cicadas as a really interesting and sort of a unique biological phenomenon that was really interesting to see during the emergence year. But I started to think of them as essentially a storage mechanism in the ecosystem where nutrients are concentrated into insect bodies, which are then released in this resource pulse every 13 or 17 years, depending on the species. And this sort of went against conventional wisdom, which often said that insect bodies are a relatively small part of ecosystem processes. This suggested like, in some cases, insect bodies could actually be a pretty important nutrient pool in the ecosystem. What I wanted to know next was, does it affect plants? So, you know, oftentimes that's the first question that people ask is, does this fertilization actually affect plants? I looked at this in a forb at first. This is an understory herbaceous plant, the American bellflower, which was pretty common at one of my sites in Virginia at Mountain Lake Biological Station. I did similar experiments where individual plants were fertilized with dead cicadas and others were kept as controls, and observed that the nitrogen in the leaves of these plants increased. So the foliage nitrogen increased. And then I did stable isotope analysis on those leaves to show that the stable isotope signature was indicative of the plants taking up nitrogen from a higher trophic level source. So it wasn't organic nitrogen, typically plant-derived organic nitrogen or plant-derived organic nitrogen from the soil. It was likely to be from an animal source, a higher trophic level. Those plants also produced slightly larger seeds and were slightly larger. Their own biomass was slightly larger. All of these things sort of together suggested that there was a fertilization effect on these plants. Taking that one step further, I wanted to know if there was any cost to this fertilization. And so that summer, 2004, and actually the subsequent summer, 2005, I measured rates of herbivory on cicada fertilized plants and was able to see that both mammalian and insect herbivores seem to prefer these fertilized plants, which actually was a very surprising finding to me. The plants don't look different after you do this manipulation. It's the kind of thing you have to measure carefully and have a lot of replication to see. But amazingly enough, both the insects and I think largely the deer in this community seem to recognize which plants were fertilized, which plants had very slightly higher nitrogen concentrations in their leaves, maybe are growing faster or taller, and preferentially ate the ones that were fertilized. In ecology, one of the questions we ask a lot is, are populations regulated from the bottom up or the top down, which is sort of a shorthand way of saying, what regulates the density of a particular group? Is it resources from the bottom up, or is it enemies, consumers from the top down that eat it? Both of them could be important, and this is a classic question in ecology. What's more important, the resources or the things that eat you? And this, oftentimes, I think after many years of studying this question, ecologists have come to the general conclusion that it's both. Both those things act in balance, both bottom up and top down factors are important. What this study made me think about was that oftentimes we're asking, what's the relative importance of bottom up or top down effects? But maybe we should be asking, do they happen in a temporal sequence? Instead of asking, is it bottom up or top down, perhaps it's bottom up at first and then top down later. So instead of thinking about them as alternative hypotheses, hypothesis 1 and hypothesis 2, we should think about them as parts of a sequential hypothesis, a time point 1 and a time point 2. In the case of periodical cicada fertilization, there is a pulsed resource which fertilizes the plants, and then a subsequent cost of increased herbivory at a later time point. And they're separated in time. And that separation in time seems important to me. So to go back to this large diagram, I think there are indirect effects of cicadas on plants and herbivores mediated through this below ground pathway. And we also see, I guess I'll just mention briefly, we also see these kinds of effects in aquatic ecosystems. So just to briefly mention that cicada inputs also affect aquatic systems in pretty interesting, in some ways similar, in other ways slightly different ways. So to conclude part 1, I think periodical cicadas are pulsed resources and that they allow us to see the structure of species interactions in communities. They serve as a link between above ground and below ground components of ecosystems in ways that are interesting for ecologists to try and link together. And this is one of those places where I wanted to make sure I stopped and asked for questions, if anyone has any questions.
There's a couple of questions in the chat, and the first one is from Pete about the nymphs. Pete, do you want to unmute and ask it? The first instars?
I'm scanning the little boxes to see Pete.
I will ask it because I don't see, I only see text. I don't see little boxes, but
okay.
But Pete is a bird guy, and he wanted to know whether
cool.
the first nymphs emerging from eggs in August form another, are they another form of a food pulse to a different trophic level?
That's a great question. We don't know very much about that first instar that goes into the ground. It seems that a lot of them get eaten by ants and other small litter-dwelling arthropods. We know that there's substantial mortality at that phase. The biomass of the insects is so much lower at that stage, but they're very numerous. So if I had to speculate, I would guess that, yeah, there probably is a resource pulse of some magnitude that goes into the soil, but it's even harder to see. In a lot of ways, that's a really cool question because it's almost invisible to most people. A lot of people notice the adults, and almost no one notices the nymphs. They're very subtle when they go into the ground. But the ants absolutely do notice them. Yeah, that's a good question. It would be a cool thing to study. You know, around August, when most other people have sort of forgotten that the cicadas are there, that would be a cool thing to study.
And Finn has a question about variability and patchiness. Finn, you want to ask your question?
Yes, sure. So when I kind of think about cicadas, I think about, you know, sometimes I'll see, and you kind of talked about this, you'll see one patch where there are all of these cicadas and carcasses, and then sometimes there'll just be an area where there aren't as many, and that's something that'll probably depend a lot on the terrain, the plants, the soil. So how do, especially because your experiments kind of relied on gauging what the density of cicadas could be, how do researchers kind of accurately pick plots that will allow them to calculate a cicada density? And is this a process that can be standardized so we can compare different birds or different locations?
That's a great question. You know, Google seems to think I'm interested in cicadas, so they keep sending me these news articles. And once in a while, I see these headlines that say, you know, billions and billions of cicadas. And I wonder, how do they know how many there are? Because of this patchiness, it's very, very hard to estimate the population density of cicadas. We can get a very good point estimate on the scale of a quadrat, say, by counting either emergence holes or wings or shed exuviae. There's lots of ways to estimate it on a local scale, a very local scale. But when we start trying to extrapolate those density estimates on a landscape scale or a regional or continental scale, it becomes very hard because of the spatial heterogeneity. I think the patchiness of cicada emergence is a really important feature of their life history and of their distribution. It affects predator satiation and a lot of other things. But from a practical point of view, if you're trying to figure out how to do a study on cicadas, and you're trying to estimate the density in a particular place, often the best you can do is to look at previously published studies that describe densities at that location. So that's what I would often do, is go back to older studies and figure out, like, where was the study conducted? How many cicada generations ago was it? What was the density in that spot? For a lot of my experiments, I was actually looking for places that had low cicada densities because that was my background control. And then I would add cicada carcasses to create the pulse, so a very local pulse. And those kind of low density places are surprisingly easy to find, even during a cicada emergence event. It's not that hard to find a spot where they aren't are. This results in a lot of disappointed news reporters every year because they're expecting to see them everywhere, evenly distributed across the landscape. But instead, they're quite patchy. You kind of have to go and find them. And if you're in the wrong place, you might never even notice them. You don't hear the chorus until you get close. Yeah, I think that's a really important feature of their ecology. And, you know, when we think about fertilization effects, probably leads to some heterogeneity in those kind of fertilization effects as well. Yeah, I think it's a super important and interesting feature of their ecology. I don't know if I answered your question, Finn. Did I get at it? Okay.
Yeah, that was great. Thank you.
My thought is that what a lot of reporters do is they take 350 per square meter and then multiply that by the number of square meters in a football field or an acre, and then say, you know, 1.5 million for a football field.
Right.
When there's always the bleachers, you know.
Absolutely.
Have them everywhere.
Absolutely.
Mark has a question, and then we'll let you move on.
Okay.
Yeah, thank you. This is so interesting. So I'm kind of imagining this pulse as being a little bit like dropping a stone into a pond, and you see a ripple of this perturbation out. You talked about the perturbation affecting the next stage, right? That the plants now have this higher level of nitrogen, and that leads to some negative feedback because they get more herbivory. Which you could imagine maybe there's other mechanisms of connection, and so maybe there'd be some kind of interesting looping. But just how far out can you see this perturbation, right? You know, you've got now herbivores that are eating high on the hog, so to speak.
Absolutely.
And then what's the downstream consequences of that?
That's a super interesting question, and one that I have thought a lot about, and it's hard to study. So there's a couple ways to think about it. One is, how far does it extend along a trophic chain? So it affects herbivores. Does it affect the consumers of those herbivores? That would be a really interesting question to ask. People have done some kind of amazing data analysis of birds, suggesting that bird populations may be affected in very strange and complex ways. One of these papers by Walt Koenig and Sandy Liebhold suggests that there's a transient dynamic in bird populations which actually persists for 17 years and results in lower bird densities the next emergence. It's a kind of amazing and interesting hypothesis. There's also, I'm going to talk next in part 2 about some effects that Rick and I have detected in trees, affecting their tree growth through fertilization. And one of the questions I have about this question is, will the effect that we saw in that 3-year experiment persist for 17 years? I'll talk a little bit more about that. And so that's the other way of thinking about it. How long does this effect extend in time? Is it just during the cicada summer? Do we see it, years from now? Is it a lasting impact, or is it a short-term impact? And those are really good questions. The further away we get from the direct impact, the harder it is to measure, and potentially the longer it takes.
Leslie, you have a question?
I did have a follow-up question. It actually is similar to what Mark was talking about. And I was thinking about it, like, if you think about this big resource pulse, another interesting aspect is that this pulse happens at different portions in the US across time. So there's sort of like a ripple effect. And so, and in fact, I actually worked with Walt and Sandy on that bird thing. And so that was one of the things we were able to do, is look at the sort of ripple effect of the cicada and how it impacted the birds across their range. And I was wondering if anyone had looked at that in terms of herbivory. So you could kind of look at it without worrying about the really small scale, like right here, there are no cicadas, but over here, there are lots of cicadas. You could just say, in this range, there should have been this huge fertilization pulse in this, section of the country in, 1998, and have people look to see if there's any kind of lag effects of other factors.
Yeah. So Leslie, you probably know of this work by Walt and Sandy as well. I don't know. You might have been on this as well, looking at tree ring records in places where cicadas were common and places where they weren't. And
I think they must have done that after. Yeah, no, I was not aware of that. So I'll have to check that out.
They did a comparison of cicada effects on tree growth, comparing places where cicadas were common and places where they weren't. And, we're able to detect effects on growth. It's been complicated. These tree ring studies have been difficult. Rick has done some as well, because they seem to show effects in different parts of the life cycle. So some have detected effects in the emergence year, some have detected effects before the emergence year, and some have detected effects after the emergence year. So we don't quite know when they are having these effects. I'll talk a little bit. I don't know if I don't know how much we'll get to, but in the experiment that Rick and I did recently, we detected effects of fertilization in the first year, but we didn't detect any effects of herbivory.
One thing I was just
go ahead.
I was just thinking. I know that we have to just really quickly. I was just thinking that with herbarium specimens now, they're actually looking at herbivory. So you could go back and look at herbarium specimens and see if there's. Anyway, just a thought. Sorry,
that's super cool. That's super cool.
I think we should press on and let Louie get through as much as he can of his talk, and then people have to leave, they can, and other people can stay and ask questions.
It's good. This is actually this is the part that I feel like is often dropped at the end of these seminars. So I'm actually glad we have time to do the question part, because that's the fun part anyway. Okay, so I will try and move through part 2 and see if there's time. So in part 2, I'm going to be talking about these 2 schools of ecological thought. So I think that one school of ecological thought is focused on species interactions, the distribution and abundance of organisms. It's an organismal perspective of ecology. But there's another school of ecological thought focused on the transformation and flux of energy and matter, this ecosystem perspective. And they are often surprisingly separate schools of thought, because they use different currencies to measure and assess nature. But I think cicadas help bridge that gap. And so the question in this section is, what's the relationship between cicadas and tree growth? This would seem like a relatively straightforward question to answer. We know some general mechanisms, herbivory and fertilization. Herbivory seems generally across plant-insect interactions to have a negative effect on plant growth. And fertilization generally has a positive effect on plant growth. And so we have pretty strong a priori expectations. However, in plant-insect interactions, we often study pulsed herbivory in the context of insect outbreaks. When they become really bad, either in agricultural or natural systems, their herbivory is quite intense, but of short duration. And that's the kind of pulsed herbivory we often focus on. And we often focus on chronic fertilization. So we study areas of different intrinsic productivity, higher nutrient availability in the soil. Cicadas flip this script. They present us with a case of chronic herbivory, long-term, below-ground, low intensity but long duration herbivory, and pulsed fertilization. They present fertilizer nutrients to the ecosystem in a very pulsed way. So our first attempt, and this is work I did with Rick Carbon in 2009, was to look at tree rings. And we went to several sites where cicadas were common, measured the density of cicada emergence under each individual tree with a quadrat, and also took a tree core to go back in time 17 years and ask, those cicadas were under that tree for the last 17 years, how have they been growing for the last 17 years? And then we took the average of their growth increment over those 17 years. The expectation was that there should be a negative effect of cicada herbivory. The higher the density of cicadas that were feeding on that tree, the slower that tree should have been growing for the last 17 years. What we saw instead was a hump-shaped relationship between tree growth and cicada density. And at first, this was a bit of a head scratcher. How could that possibly be? You know, this herbivory, the negative effects of herbivory seem very general and very common across plant-insect interactions. And I was just interested in quantifying it, not expecting to see something else. What I hadn't anticipated was the possibility that this relationship is actually being driven by two distinct processes. One possibility is that there is a negative effect of cicada herbivory, but also laid on top of it, a second process by which cicadas are preferentially ovipositing on faster growing trees, or somewhat more magically, trees that have a better prospect of growing faster for the next 17 years. So they're anticipating the growth conditions of their host plant over a scale of 17 years. And if there are both of these processes going on, the result might be a hump-shaped relationship. So if cicadas are placing higher densities on faster growing trees, but also higher densities of cicadas slow down tree growth, it could result in this hump-shaped relationship. Well, what's the evidence for this? We know that cicadas are patchy in space, and when we looked very carefully at our cicada emergence data, we saw that the part of the quadrat that was close to the tree actually had a higher density of cicadas than the part of the quadrat that's far away from the tree, suggesting that they prefer to be in places with a high root density. I also looked at the rate of oviposition and correlated it with the canopy cover above the trees. This was an experiment I did where I planted about 100 oak seedlings and then used a hemispherical camera to measure how open was the canopy above those trees. And these were small trees, you know, probably less than 5 feet tall, probably less than 4 feet tall. They differed in canopy openness, and canopy openness was strongly correlated to the density of cicada oviposition on the trees. The cicadas seem to be preferring to oviposit on the seedlings that had more open canopy above them, more space to grow, essentially. Somewhat surprisingly, when I looked really closely at the cicada oviposition scars on those trees, they were preferentially ovipositing on the west side of the trees. And the western side of the tree is the part of the tree that's exposed to the afternoon sun. So taken together, this suggested to me that cicadas like to oviposit in the sun, and they're most active in the afternoon, and that's when they are doing their oviposition. So to try and put that together, I think that there are several processes that affect tree growth. One is the light environment over the tree. The more photosynthesis the tree can do over a long time scale, the faster it'll grow over a very long time scale. It's a pretty crazy world out there. And if you're trying to predict tree growth over 17 years, a surprisingly good predictor of that is canopy openness. The tree growth rates, high densities of roots seem to have a positive effect on nymphal density. So the higher the root density, the more nymphs that tree can support. High light environments also seem to lead to higher densities of cicada eggs, higher oviposition densities, because the cicada females are choosing wisely in terms of habitat selection. They're choosing trees that have more light above them. Nymphal densities have a negative effect on tree growth rates. So the more nymphs are feeding on the tree, the slower the tree should grow. That's sort of the a priori prediction. But then add to that the resource pulse effect. Higher cicada densities, carcass densities, should increase tree growth rates through fertilization. All of which is to say that what I thought was going to be a simple question might actually be a more complicated question. There are several biological, ecological processes going on that link cicadas to tree growth, both through fertilization, herbivory, and unexpectedly through cicada behavior, habitat selection. So if we try and approach this as an experiment and do a two-factor experiment manipulating herbivory and fertilization, what are the combined effects of herbivory and fertilization? As Rick would say, this calls for an experiment. So I was telling Rick how I was getting all nostalgic about having studied cicadas for 17 years. And Rick reminded me that he first studied cicadas in 1977, which is the year I was born. So he's hoping to get 4 cicada generations in his academic career. He's been doing this for a while. We decided to do an experiment in Kansas with 100 sycamore trees, which we planted out in a field. And we did the simplest possible experiment we could imagine, a 2x2 factorial. We randomly assigned 25 of the trees to receive fertilization, 25 to receive herbivory, 25 of them would receive both herbivory and fertilization, and 25 would receive neither. So it's a factorial herbivory by fertilization experiment. And then I guess to cut to the chase, we saw strong effects of fertilization on tree growth. The trees that were fertilized grew measurably larger and faster in the first year. Surprisingly, we did not see significant effects of herbivory. So one of the questions that this raises for me is, will these effects amplify or attenuate over time? The argument for attenuation is that the world is noisy and a lot of things are going to happen over the next 17 years. This initial perturbation to tree size may just get washed out with the vagaries of time, and the trees will even out and this effect will dissipate. The alternative expectation is that they may amplify over time. So we know that light competition among plants is strongly asymmetric. The taller tree gets the light, the shorter tree gets less, and that effect compounds. It's a positive feedback. So the taller tree gets taller, the shorter tree is progressively more shaded. So one alternative expectation is that this very small perturbation in year 1 might set into course a positive feedback by which the taller trees now have the means of production, and they get taller and taller, and the shaded trees end up getting outcompeted. And that's one of the things I'm interested to see. Our experiment ran for 3 years. The trees are still out in Kansas. We hope to go back every few years and see if that effect amplifies or attenuates. The other surprise was that there wasn't a significant effect on tree growth through herbivory. We expected to see one in the first 3 years because these tree ring studies all showed effects within the first 3 years. One possibility is that the effects of cicada herbivory might be context dependent. It has always struck me that one of the biggest things cicadas do is to make roots leaky. They take in a lot of xylem fluid and excrete a lot of dilute urine, and that essentially makes the tree less efficient at gathering water. And it seems possible that in wet environments, that's not a big cost, but in dry conditions, that might be a big cost. Kansas is a place that has periodic droughts, and I'm wondering if the effects of herbivory are only manifest during drought conditions. Oh my goodness, I noticed it is 10:32, and I want to apologize for anyone that needs to run. I have been there many times, and if you need to go somewhere, if you are juggling lots of things, you should go and do what you need to do. Thanks for coming. Sorry for running over. I'm also happy to continue for those of you who are willing to stick it out. All right.
Since we're not in a room and the next class is not coming in, let's just proceed, and whoever needs to go can go.
Awesome. Fantastic.
You're getting some appeals to continue in the chat.
Okay, cool. Thanks. Yeah, I applaud everyone that is doing this while juggling lunch and kids, and like, I've been there. Okay. Okay, so to conclude part 2, I think the relationship between periodical cicadas and tree growth is surprisingly complicated and requires a combination of behavioral, population, community, and ecosystem approaches. There are a lot of processes that are mediating that interaction. It seems likely that the cicadas and the trees have a reciprocal interaction. The cicadas are affecting the trees in a number of ways, but also the trees are strongly affecting the cicadas in a number of ways. And then third, the effects of chronic herbivory and pulsed fertilization may differ in time. Chronic herbivory effects may be context dependent, depending on drought conditions, and fertilization effects could either amplify or attenuate, depending on how those dynamics work out. And I guess those questions remain out there. And I can pause briefly for questions, or I can roll on to the very short part 3. What would you prefer?
Mark had a question.
Go ahead, sorry.
Yeah, I had a quick question. So you have the ovipositioning effects and I guess herbivory in the end as being this independent of the carcass load. And, and it made me wonder, of course, I guess they fly away, but is there any correlation between carcass density and oviposition?
That's a great question. I think that there is likely to be, if we collected those data, I would guess that there is a correlation there, especially on a landscape scale, because there are so many places that have neither, that are zeros for oviposition and fertilization. So on a big scale, there's almost certainly some correlation there. And I think a lot of them die near places where they oviposit. And so I think those are actually linked through the cicada life history themselves. So it actually seems quite likely that we would see both fertilization and herbivory in the same places, in the same trees, at least in a statistical sense. The caveat is that they're not perfectly correlated because they do move around a fair bit. Oftentimes they will chorus in a relatively small patch and then disperse for oviposition. So I think that if we were very careful about measuring it, and in fact, some people have done these kinds of studies, I think we would see that the carcass densities are not perfectly overlapping with the oviposition densities. So there may be some winners and losers, which is to say some trees are getting just the positive effects and other trees are getting just the negative effects of cicadas. Yeah, this sort of goes along with Finn's question about the heterogeneity. I think cicadas have an interesting interaction with heterogeneity, forest heterogeneity. They prefer to oviposit in these sunlit patches, often fast-growing, secondary growth, relatively exposed trees on forest edges or parts of the forest that are growing faster. So I think their habitat selection is non-random and probably a pretty important driver of spatial distributions of oviposition. Cool, cool, cool. That's why they love the suburbs, totally. Okay, maybe I'll do the very short part 3 because it's mostly just me pontificating on my soapbox, and then I'm happy to take questions after that. So I feel like the third thing I've learned from periodical cicadas is the importance of this temporally explicit perspective. There are a lot of ways in which cicadas invoke this time component. One is the importance of sequential hypotheses for top-down and bottom-up. Another is the synchronized element of consumer responses. If we took that same cicada pulse and spread it out over 17 years, I don't think we would see the kinds of dynamics that we see. What we see is a consequence of their pulsed nature, not their sheer absolute magnitude. I guess just thinking about cicadas from this perspective, I realized that cicadas were a start for me that forced me to think about time. And I've taken that perspective to do other things. So, for better or worse, cicadas aren't really the only thing I study now. They're probably not even the main thing I study, but they're always going to be the first thing I studied. And they're always going to be an important part of the way I think about ecology. So now I study pulsed subsidies on islands, and I study the timing of monarch milkweed interactions. I study other kinds of insect interactions that seem strongly seasonal. And, I do some modeling of phenology. And all of those things, I can draw a link back to cicadas in terms of how they made me think about ecology. So I have come to this perspective that it's important for us to look at ecology in a more temporally explicit way. And I sometimes have a hard time describing what that temporally explicit perspective means. And I've decided that my best hope is to use an analogy. And so here's my analogy. It revolves around paintings of horses. And I'm going to make the controversial claim that this is not a horse. It is, of course, a painting of a horse. It's a representation of a horse, and it's not even a very good representation of a horse. If you're a horse person, you look at that and you think, that is a ridiculous-looking horse. This is a horse that was painted prior to 1870. It's painted in what's called the hobby horse position, in which the front legs are extended forward and the hind legs are extended backwards. And if you go to a museum, I know you have a lot in the DC area, you go to a museum, look at their fine art gallery of all paintings of horses prior to 1870. They are all painted. If they're running, they're painted in this hobby horse position. Front legs extended forward, back legs extended back. Just doesn't look natural to the modern eye. But this is the way we thought horses ran. This is our best guess at realism. And these are people who spent a lot of time around horses. They saw horses probably more than we do. But they did not have the ability to see how horses actually move. Does it matter how horses actually move? Well, it did to some people. Leland Stanford was so interested in how horses ran, he commissioned a photographer, Edward Muybridge, to take photos of horses in motion. He was lucky enough to have a private horse track, and Muybridge set up cameras all along the track and had the horse trip them sequentially to take photos of horses in motion. So this wasn't an easy thing to do, but wanting to know how horses actually ran motivated Stanford and Muybridge to do this experiment. So I'm going to argue that this is not a horse either. It's also a representation of a horse, but it's a much better representation of a horse because it includes the dimension of time. It essentially does that by taking lots of slices of time and putting them one after the other. And that creates the impression of movement that allows us to see how things actually are. All right, so to bring the analogy all the way around, I'm going to argue that this is not an ecological community, and it's not even a very good representation of an ecological community. We know that real ecological communities are more complicated than these kind of node and arrow models suggest. Real ecological communities involve, movements across habitat boundaries and reticulate connections and self-connections and connections of different strength. But even this diagram misses out on the element of time. When we add in the element of time and we allow connections to blink in and out, to change in their intensity and duration, when we allow organisms to themselves change, over ontogeny, we have something closer to a temporally explicit ecology. And that's the kind of thing that I'm slowly trying to edge my way around, try to understand how is it that communities are actually changing in time? How do interactions of different dynamics interact with each other in a temporally explicit way, to get something closer to a moving horse picture of ecology. So that's where I'm hoping to end. And apologies for running longer than I intended. I really enjoyed the questions and comments, and I'm very happy to stick around and answer questions for as long as folks are willing to listen to me.
Thanks so much, Louie. We do have
Thank you.
We do have a few pending questions.
Cool.
And Christopher asked if there were some trees whose roots the cicadas avoid.
This is a great question, and I don't think we know. This is like the classic ecology answer. I don't think we know that much about how cicadas operate below ground, but it does seem evident that they have very wide host range below ground. So they can't oviposit in sap-bearing trees. They can't oviposit. They can't lay their eggs in pines and conifers and things that have defensive sap. It just gums up the works, and the eggs don't survive. The ovipositor doesn't work. But they can feed on the roots of conifers, and they can feed on the roots of grasses, and they can feed on the roots of seemingly a very wide range of host plants. So we see oftentimes when trees overhang a meadow, for example, the cicadas will be feeding on the meadow plants and then emerge during the emergence event. Or if a woodlot is cut down, there are no more trees there. If the cicadas were already established on the roots of grasses or forbs, they seem to do just fine. They're also, I guess, from a physiological basis, they're feeding on xylem fluid, which is relatively undifferentiated, relatively unspecialized fluid in the plant. And so it makes sense that they would have relatively wide host ranges, but they don't seem to be able to oviposit in trees that are well defended.
Do we have other people?
Ben and Mark have their hands up.
Okay.
Oh yeah.
Our best customers always. Ben?
Well, thank you for lumping me in with Dr. Rose. That's quite a wonderful question group to be in. So thank you for the really interesting and relevant talk. And I think that you and your work do a great job of showing that the cicadas themselves and their carcasses can create these resource pulses that impact plants and other herbivores in their ecosystems. Personally, I think that these pulses provide a major opportunity for local populations. So do you think that other organisms have evolved to take advantage of these cicada emergence events?
That's an interesting question. I mean, the emergence events and the dynamics that are going to happen, in your area this summer, are super interesting, super seemingly important during the emergent summer. But then, 16 more years will go by where they don't happen. And so I think in a lot of ways, it makes it hard for organisms to adapt to capitalize on the cicada resource pulse. I mean, I think that is likely to be, from the perspective of the cicada, largely a feature and not a bug of the prolonged development. So in a lot of ways, I think adapting, evolving to capitalize on that resource pulse is challenging, for pretty good reasons. People often point to this fungus, Massospora cicadina, which is a specialist pathogen of cicadas. It's often described as the only natural enemy of cicadas that has evolved the ability to to essentially chase their populations through time. It's dormant in the soil. It waits for them to come out. There's no other consumer population that seems to be able to wait for them in that way. So yes, I mean, I suspect it does have an effect on selection. I suspect it does affect evolution. But then the question is like, sort of the same as the tree growth question, does that effect persist? Does it attenuate? Does it amplify? And those are the kinds of interesting temporal questions that I think we don't know yet. I mean, I think these really interesting studies with bird dynamics are super interesting because the transient dynamics that follow a strong perturbation like this are probably a big part of the signal, the transient dynamics that happen after a large perturbation. Yeah, I would love to see more thinking about sort of the evolutionary impacts of cicadas. That'd be cool.
Mark?
So I will return the compliment, Finn, and say that I hope that I never lose your level of curiosity, because you know that's what I continue to aspire to. So I'm happy to be in your company. So thank you for this great talk. It was super accessible even to a molecular biologist like me. So way back when I took ecology in school, I remember learning about the Isle Royale, you know, foxes and rabbits, and maybe that's not even true, but it did bring in this temporal aspect where you'd get, you know, population crashes and booms and between the predator and the prey, I guess. So, but it just, I'm wondering about other examples where now when you bring in this temporal relationship with different kinds of feedback, you can get into really weird cycles, right?
Mm-hmm.
And so I'm really curious about examples where you have really strange cycles that happen due to that. And so this is not necessarily about cicadas, but maybe other things you study.
Yeah, no, this is a cool question. In a lot of ways, I think cicadas are an awful species to look at population dynamics. It's just so hard to see more than a few generations. I made a trip to Raccoon Grove in Illinois, which is the site where the highest density of cicadas was recorded. I think it was in 1954, maybe 1956, somewhere in the middle of the 20th century, the mid 1900s. That's where the 350 cicadas per square meter was recorded in a floodplain. And I got some old maps and figured out, you know, where they were when they recorded that. And I was there for the next cicada emergence, you know, in my academic career, which I think was, gosh, that would have been what, at least three generations removed. Quick 17-year math. Yeah.
88 maybe?
No, it must have been 4 generations removed. It was several generations distant from the Raccoon Grove emergence. And there were almost no cicadas. So it had gone from being the highest recorded density of cicadas to being virtually absent. There were a few chorusing in the trees, but they were basically not there. And I suspect that the grove itself is protected. It's part of the forest preserve system in Illinois. It wasn't land use per se. I suspect that they disappeared as a result of the fungal pathogen. So the fungal pathogen we know builds up at high densities in high density populations. It's sexually transmitted. And so at high densities, it can transmit quite readily through a population. And I imagine what has happened at Raccoon Grove is a kind of example of a lynx and hare kind of dynamic, where essentially at high densities, the pathogen spreads efficiently, it spreads through the population and becomes a pretty effective top-down control on the population. And that we're seeing a point in the cycle now where the Massospora, the fungus, is at a high density and the cicadas are at a low density. We know that the fungus can wait 17 years. So it's not a great example, but in terms of this kind of population dynamics that we see with lynx and hare and other kinds of classic time series data, I suspect something like that is going on with cicadas, but it happens on an unfathomably long time scale that, many academic careers linked one after the other would have a hard time to see it.
But you could be working on elephant breeding, I suppose.
Yeah, exactly. Yeah.
Chris asked the classic prime number
Oh my
question
goodness.
and that is something I still like try to wrap my head around. So do you have any thoughts or wisdom about that?
Oh my goodness. This is such an interesting question, and one that has confronted evolutionists, ecologists, anyone that has heard of periodical cicadas. Once you realize that there are 7 species of periodical cicadas, 4 of which have 13-year periodicity and 3 of which have 17-year periodicity, the obvious question is, why do they have prime numbers? Is it a coincidence? Is there something special about the prime numbers? I feel like my response to this is that there are actually 3 things we want to know. First is, why do they live so darn long? Why do they have this prolonged development time? And we know some of the answers to that. One of the answers is that they're cicadas, so phylogenetic inertia. Cicadas live for a long time. They feed on xylem fluid. It's not a great resource. They grow slowly. They often live for multiple years, but not 13 or 17 years, as far as we know, not typically. So part of the answer is they live a long time because they're cicadas, but why 13 or 17, I think, is still unresolved. And then part of the question is, why do they come out all at once? If they came out every year and they lived for 13 or 17 years, we might not notice it because we'd see a small fraction of the population every year, but they're not synchronized. And so why are they synchronized? Well, again, I think we know part of the answer. Part of the answer is predator satiation is very effective. And if they dribbled out in small densities year after year, they would get taken out by birds. And in fact, we have experimental evidence that that's true. If you create a small population of cicadas, they get taken out by birds at a tremendous rate. The predators will just destroy them. They really do rely on predator satiation and that synchrony in order to be a viable species. But that's not a great answer. That's not a complete answer either. And then the third question is the one you bring up, is why prime numbers? And part of the challenge of that question is we don't know if it's chance or not. We only have 7 observations of these species, and they're not independent. So we don't know if the prime number thing is a coincidence. It could still be a coincidence. I think there are sort of 2 main branches of explanation for this. One is a version of the predator satiation, that prime numbers are better for avoiding predators than other numbers. Although I think upon inspection, that is hard to understand how that actually works. It seems like, anything longer than 2 or 3 years would be enough to avoid predators actually synchronizing to you. The other explanation I've heard is that it's an attempt to avoid the overlap between co-occurring broods. In other words, it's an attempt to minimize the risk of competition below ground. So if you have large prime numbers, 13 and 17, that only happens, the coincident timing of a neighboring brood is once every 221 years, I think. So it's quite infrequent. And that would be a viable explanation if the coincident timing of neighboring broods was really bad. And so that's actually an experiment that I'm working on with Rick right now to try and figure out how bad is it to compete below ground if you're a periodical cicada. This below ground competition might be a big part of why we see 17-year, 13-year cycles. And also there's a very strong pattern of non-overlapping cicada broods. So if you look at those brood maps that I showed earlier, they're strikingly puzzle-like. They don't overlap very much. But Chris Simon and colleagues, John Cooley, Dave Marshall, have done these amazingly high detailed maps of cicada emergences. And even at a very, very small scale, like the woodlot scale, they seem puzzle-like. They avoid overlap. So like there'll be a little gap in one brood and it'll be filled by another one. And that suggests that overlap is bad. Why is overlap bad? It might be below ground competition. So this kind of puzzle-like distribution where they seem to diligently avoid sympatry seems like it may be related to the 13, 17 year thing. So there's a whole bunch of talking without any real answers. But I think basically we don't know. Like it's a complicated question, but it's useful, I think, to recognize that that question actually has several parts. There's the prolonged development part, the synchrony part, and then the mysterious prime number part.
Mark's got a question about annual cicadas. And so how do they get around the predator, the need for predator satiation if they come out in kind of smaller numbers every year?
Great question. If you've ever tried to catch an annual cicada, you know that they're both hard to locate and hard to catch. Quite strikingly different behavior than a periodical cicada, which are so predator foolhardy that they fly into you. These annual cicadas are often, you'll hear them chorusing, so you know they're there, but they're very hard to see. They're cryptically colored. They're very hard to locate. Their call is very hard to locate. And if you approach them, they're often very flighty. So I think that they have taken a very different strategy for predator avoidance. They occur at relatively low densities, and they're very hard to catch. And so I suspect that that syndrome of traits, is so different than the satiation strategy that periodical cicadas use. So they get by essentially by being cryptic. Sort of like most other organisms, they avoid predators pretty diligently, and they're pretty good at evading them.
Do they have a better food source underground?
Oh, yeah, I don't think so. This is actually a great question. I suspect that periodical cicadas are capable of developing much faster than 13 or 17 years in terms of they don't need that long a time to develop to the biomass that they develop to. One of the things that is surprisingly hard to figure out is how long annual cicadas take to develop. We see them every year. And so oftentimes in the literature, you'll read people estimating that they develop for 3 to 5 years below ground. That's often the estimate that I've heard. But it's actually very hard to know if that's true without following individuals. My sense is that the potential to develop below ground for cicadas is faster than 13 and 17 years. I actually think annual cicadas may actually develop reasonably quickly, like within the space of a few years. The reason I think that is Rick and I have found cicadas in the ground in places where we are pretty sure they couldn't have been there for more than a decade. It was disturbed or it was not a viable habitat. And so the cicada must have developed on site within the space of relatively few years. But it's a surprisingly hard question to answer without doing the direct observation.
You need to look around a place like Fukushima or something, where there's a radioactive pulse.
Yeah, that would be interesting. That would be interesting. I mean, one of the things that I often thought about doing, before I got distracted with lots of other things, was essentially building rhizospheres to observe cicadas below ground. It would be one of those like long-term, probably foolish things to do, but it would be super fun to try to establish cicadas on containerized plants with a visible panel where you could essentially watch the nymph develop and just do the direct observation. I mean, it's kind of the first thing to try, but it would be super fun. Y'all should do it.
It's like a giant ant
Exactly.
Louie, I have a question about xylem fluid and nitrogen and mycorrhizae. And my idea had always been that it wasn't going to be a big deal for the trees because xylem is pretty much mostly water, and it's, if there's enough rain, it's cheap. It's not like taking the soda pop in the phloem.
Right.
plants haven't paid for it in particular yet. But then I
Right.
started thinking about the fact that the plants are taking nitrogen up as they are taking up the water, sometimes with the help of the mycorrhizae, and that the cicadas are then kind of basically stealing some of the nitrogen that they're taking up.
Yeah.
So that it could in fact be a hit for the trees if there's not a lot of nitrogen in the soil, or if the mycorrhizae aren't around or whatever. And so I guess I'm asking how much mycorrhizae have to, a good mycorrhizal network has to do with this whole thing. And if the trees do resent the siphoning off of nitrogen, or is that sort of like a carbon resource from the sun sort of thing?
I mean, the short answer is I don't know, but it's a great question. I mean, the mycorrhizae aspect in particular seems like a really interesting wrinkle where both nitrogen and phosphorus seem like interesting limiting resources. I don't know. I mean, my impression is similar to yours, that the xylem fluid is so dilute that it's mostly likely to incur water stress for the tree. And not sort of nutrient limitation, that you can get more xylem fluid relatively easily most years. But it bears an experiment. Like, it would be an interesting thing to try to quantify those fluxes and the role of mycorrhizae if you could. This isn't this is not an answer to your question, but I've often wondered if one of the impacts of cicadas is actually to create something like hydraulic lift. So in a lot of deep-rooted plants, they will take up water from deep roots, and then when they have more water than they need, they will release it at, I think at night, into shallow soil layers. And plants do this sort of naturally, but it seems like cicadas may exaggerate this hydraulic lift. Essentially, plants are working hard to pull up groundwater, and then the cicadas, which are relatively shallow, are releasing a lot of that excess water as urine. And so it would be interesting to do some back of the envelope calculations to figure out if that is a meaningful transfer of soil water to shallow soil layers. I guess the impact could potentially be changing plant community water dynamics for shallow-rooted understory plants. I've often thought that this is probably a bigger deal in arid ecosystems. So like in desert ecosystems where there are some cicadas that occur in desert riparian zones. And it seems like this shift from deep water stores to shallow water could actually make it possible for plants to grow in some of these places if the quantities were large enough. But you know, it's just one of these things that I haven't quite figured out where or how to test. Some back of the envelope calculations might be the thing to do first, but yeah. Martha, that's a great question. I wish I knew the answer, but yeah, that's yet another thing we don't know.
I do see another question from Chris here about the loss of the chestnut trees, and whether the loss of the American chestnut affected the cicada populations.
This is a great question as well. You know, I feel like one of the things that has been fun about thinking about cicadas this year is that, you know, it's been a tough year, and it's fun to think about a system that seems separate from human concerns. But every once in a while, these human concerns kind of nose their way even into periodical cicadas. You know, I don't want to be a downer, but one of the things that I have noticed, partly because I grew up in West Virginia, but then now I go back somewhat infrequently, you know, every few years to do some research back east. At least to me, it seems like the forest has changed pretty profoundly. Because I'm not there all the time, I can see it. You know, it's been a few years, I go back and gosh, the forest seems really different. There just aren't as many, you know, the hemlocks are gone, or, you know, the other species are less common, or there are more dead snags. Those changes seem pretty big to me. because I don't see them every day. And the loss of the chestnut trees was certainly one of those very big changes in those forests that it happened, of course, well before my time, but the sort of bigger, I guess the bigger issue is the shifting baseline, where our own basis for understanding these forests is so limited. You know, my short career as an ecologist is not enough time to see all the changes that have happened. But even within my short career as a biologist, I've seen pretty big changes. So, I mean, I suspect the loss of the chestnut trees has had pretty big effects, but I don't know what they might be. People describe those chestnut forests as being sort of canopy-like, and sort of creating this like cathedral-like effect of a high canopy, and potentially you could speculate that sort of the lack of secondary growth was actually bad for cicadas. These mature primary growth forests, relatively dark, slow-growing, mature trees might have actually been not so great for cicadas. They might do pretty well in the kind of scrubby, forest edge, fragmented kind of habitats that we create. But I don't think we know terribly well. Interestingly, I'm not going to make it out to the cicadas this year, but Rick is, and he is going to work on the experiment that we have out at Blandy this summer. But his main goal is to actually recensus a population that was originally censused prior to the chestnut blight, and then again recensused in 1977. When Rick was, I think, he told me, I think 22, 24, something like that, when he was just starting in grad school himself. And so he wants to go back and recensus it and see how the forest community has changed. So he'll be there driving around this summer and trying to census a forest that, was censused prior to chestnut blight, again in the late 70s, and then again now, 44 years later.
We should definitely try to hook up with him.
Yeah, I was about
Absolutely.
to say, let Rick know if he needs any help. There are people here who will happily do so.
I think he's he's excited. He's going to meet his daughter out there, who's now a graduate student in ecology, and then they're going to do that forest transect survey together.
Oh, fun.
Awesome.
Super cool.
John, you want to ask your question?
Sure, just a quick one. So just going back to the plant responses that are so confusing about so one of the things we've kind of wondered about is whether, the year, like right now, as these fourth instars are basically done feeding and then they're going to emerge, next year, will there be sort of a release from underground herbivory that would be replaced by a bunch of nymphs that maybe aren't even on the roots the next year? It might take a while before they get there. And like it's such a minimal one. And I'm vaguely remembering that some people have reported a pulse of growth the year after but then you also have the nitrogen inputs to the soil. So it's like, it's hard to sort of distinguish those two things, because we measured it in some understory beech trees and saw a big jump in biomass production the year after. And but we really couldn't guess. So I don't know what your thoughts are on that.
Yeah, I think that's why that has been such a difficult question. These tree ring studies have found differing years of effect. And they offer plausible explanations. So studies that find a reduction in tree growth the year before emergence speculate that, well, the nymphs are the largest at that point, and so they're having the strongest draw on tree resources, and so that's why you see it the year before. And then studies that show effects the year of emergence say, well, that's the year that the first instar nymphs are reattaching to tree roots. And it's thought that that reattachment process is pretty messy. They damage a lot of fine root structures. And so they're causing plant stress because there's a high density of sort of sloppy root feeders that are causing a lot of plant damage. And so those are both plausible explanations. And those tree ring studies legitimately find effects, but in different years, it makes it hard. I mean, part of what I think is likely to be happening is that it's context dependent, that because they're feeding on xylem fluid and xylem fluid's not particularly limiting, it probably depends a lot on the weather conditions in those different years. But we had hoped that we could resolve this experimentally in the Kansas experiment, but we didn't see any meaningful effects of herbivory in the first 3 years. And so it might be one of these exceptions that proves the rule that cicada herbivory doesn't have a huge effect on trees, except under certain conditions or under particular cases. I think Rick would disagree. He still thinks that there are big effects, but we just don't know when or where to look for them per se. It seems, I mean, my gut tells me there are probably effects, but I don't know when or where to find them.
Lots of tolerance possibilities still, right? So plants are pretty amazingly tolerant of a lot of
Absolutely. Yeah. I mean, I think that is also quite a reasonable expectation. We're so unused to dealing with this chronic herbivory situation as opposed to the pulsed herbivory situation, and they're taking such a small amount. Like we were saying, cicada biomass probably doesn't need 13 or 17 years to develop, and I've heard some speculation that they are not feeding very actively for part of that time. They only need a small fraction of that time to develop to the size that they get to. So they might be spending a lot of time underground just taking, maintenance amounts of resources from those trees. But again, we don't have great direct observations of it. It would be cool just to record the rate of biomass accumulation if you could see the nymphs develop, even if it's just over the first 5 years or so. How quickly are the nymphs growing, and how actively are they feeding during that time? That would be useful to know.
It shouldn't be that hard. I mean, I know Chris Simon has some protocols worked out, but it shouldn't be that hard to dig them up at every May. And, on Mother's Day, go outside and dig up under your maple tree and weigh 20.
I love it. The Mother's Day cicada census, like a Christmas bird count for cicadas.
It's starting here and now.
I have tried digging up cicadas on a few occasions, and I have not been successful. It's very hard to do, especially when they're little. When they're big, you see them, but when they're little, they just kind of get smashed up in the soil, and it's very hard to identify them on roots. I know people have done it, but it seems to be a pretty chancy enterprise and hard to do quantitatively. Even in places where I'm pretty sure they're there at high densities, finding out the depth they're at and knowing that you're getting, I don't know if anyone has had success doing this, but I've tried it on my own without a lot of success. I've tried it with Rick a few times without a lot of success. It seems really hard to do.
Yeah, honestly, I've never seen him except for the fifths, but I was looking at Chris Simon's website last year, and she had a protocol, and you go dig them up and you put it out on the tarp and you sort them out, and it takes an hour, and you make a little graph.
That's awesome. I mean, Chris and colleagues have done pretty amazing things with these cicadas over the years. So if anyone knows how to do it, I would guess they've figured it out. I
bet it has something to do with the soil texture as well.
I suspect that's really true. Some of these places just digging is hard. And digging to the root, the rhizosphere is very difficult in some of these places. I've actually wondered if there are any molecular approaches that could let us know if cicadas are on a tree. Like, could you take a bulk soil sample and then use a molecular approach, almost an eDNA approach, to see if there's any trace of cicadas in a bulk soil sample? I've wondered about a bunch of these things. Is there enough transfer of cicada DNA into the xylem? Could you measure it in the tree itself? I don't know. I feel like there's a whole next generation of cicada type experiments that might take advantage of these molecular approaches if we could figure them out.
Yeah, some of the problem with the cicada, looking for the cicada signature in the soil, would have to do with the ground up dead cicadas from the last generation and, little bits of chitin and stuff like that.
Yeah, that's possible, right?
I've been doing my cicada cookery experiments, and so I have been going over to my neighbor's house and picking up their flagstones and collecting their hit a brick wall nymphs. And I cooked some up the other day, and on my little neighborhood listserv post, I said, you can taste the terroir of the magnolia tree. And it occurred to me that it is possible that the different trees, I don't know if there is, in fact, at that point, any substance from the trees in there, but I wonder if those of us with discriminating palates could tell the magnolia cicada from the oak cicada.
That would be a great thing to do, both quantitatively and as a sort of taste panel. Could you discern through any molecular or analytical chemistry approach, and can you discern it on your palate what the host plant might have been?
Yeah, I mean, if they took pine trees or something like that.
Absolutely.
That ought to taste a little more resiny. Honestly, it might be a little bit better than the rather blah magnolia cicadas.
It's a good sign that you're seeing the nymphs though under the flagstones. That's
oh my God. I mean, we're not just seeing the nymphs. I mean, we're within days. I mean,
yeah, it's happening.
Been up since for 3 weeks or a month. I mean, in pretty large numbers. So and John has been doing this in his yard, but picking up the flagstones, we see 20 or 30 under a 2 foot by 1 foot flagstone, and they come up and they hit the wall. They go a little sideways and then they go, well.
Yeah.
We had like 30 in an 8-inch square stone. There were 30 nymphs. It's like really high densities, I think.
That's terrific. I'm actually glad to hear that, because I feel like the last few that I've gone out to look at have been sort of disappointing in their scale. I went back, my first cicada brood was actually 2002, brood 8. And so I went back there in 2019. It's sort of in Pennsylvania. And it was sort of disappointing. It seemed less dense than I remembered it in 2002. I don't know if that's a trend or not. It's not enough data. But I'm glad to hear that they seem like they're at high densities in the DC area.
We had a lot come out in 2017, so we were kind of worried that was going to dampen it a little.
Right.
2017 was pretty good. Like, so there were local choruses forming in 2017.
Right, I heard that. Yeah.
I heard them on the C&O Canal, and your places you sampled along the Potomac were really good. So there were a lot of 2017 ones, and then there were a reasonable number last year that came out 1 year early. So even from the same brood. So I was kind of wondering, but it looks like it's going to be dynamic.
That was it's not my place. Yeah. Here's the Wackler's.
Wow, that's amazing.
That's the Wackler's flagstone.
That's impressive.
Yep.
Yeah, that seems like a good sign. I mean, I think that the stragglers are really interesting, and I guess I wouldn't be surprised if the incidence of stragglers is changing with the changing conditions in the world. But that's super interesting. It'll be interesting to see what happens this year.
Zoe, have you ever met Louie? Do you guys know each other?
I don't believe we have met.
Would you care to introduce yourself?
Hi, I'm a postdoc working with John and Martha this year to look at how the cicadas are affecting local food webs, insect and bird food webs.
Super cool. I saw your comment earlier about the bird. Let me see, scrolling through the chat. I love the chat function.
Yeah, we were chatting with Pete about what the birds would be doing and why those numerical responses might be, and he seemed to think it was an artifact of how the observations are focused on the song, and there's much less need to be territorial if food is flying straight into your face all day.
Yeah, I think that's super interesting, and it sounds like Leslie might have some insight into this as well. I mean, I know that paper talks a bit about the potential for observation bias during cicada years, because so many of the surveys are auditory. If you're in the right place, you can't hear a thing during a cicada emergence. And I thought that might be an explanation for it. But they had some sort of rebuttal to that argument.
Yeah, their argument was more that the cicada chorusing would mask the sounds, which, yeah, they did account for decently by having volunteers record how many cicadas they heard, but it wouldn't necessarily account for birds flying out of areas to go to cicadas and singing.
Right. Yep, that makes a lot of sense to me. I mean, I think it doesn't make sense that you would forage far during a cicada emergence. It might be more of a localized patch dynamic. You go to one place and not that far away. Yeah. Yeah.
So part of what we're looking at, and I think probably, John, I don't know if you have told Louie about what the big picture of our project is, but we're interested in the effect on the oak caterpillar, the oak herbivore fauna, with the idea that maybe when the cicadas are out, a lot of the birds that ordinarily have to spend a lot of effort finding tiny little snack-sized green caterpillars can just, open their mouth and have the cicadas drop into them. And in order to look at that, last year we put out clay caterpillars and census them for bird beak marks every week for the whole summer. And we surveyed herbivory, so we know how much oak tissue we lost. And we did a decent job, I think, of looking at, I mean, we followed the trajectory of the LEPs, but and did some censuses. And then this year, our predictions, which are a moving target, are that when we put the clay caterpillars out this year, during the cicada emergence, we are thinking that we will have fewer bird beak marks on the caterpillars, and we will have more caterpillars surviving and higher levels of herbivory on the oaks. And theoretically then higher parasitism loads the following year, because more caterpillars are surviving with their parasitoids in them that will seed the population the following year. But that later in the season, since we did it for 12 or 13 weeks last year, after the cicadas are gone, then we'll go back to a similar beak situation. There's a ton of things that we don't know. We don't know if the cicada, the birds that are, I mean, there's sort of the generalist birds that are going to be taking any old thing, and then there's some other kinds of little warblers that really are little caterpillars, gleaning under the leaf specialists, and maybe they won't change, and but the cuckoos and the blue jays will. And so, we're going to see. Did I, John and Zoe, did I explain that about right?
We're excited to include that temporal component that you were mentioning earlier in your talk.
Yeah, that's super cool. I love that hypothesis. It's the apparent mutualism expectation, and I think that's super cool. I made a half-hearted attempt to try and answer a similar question years ago, and just didn't get very far with it. But I think it's very plausible that predators will focus on cicadas and ignore a lot of other herbivores. That seems very plausible to me. And that sounds like the artificial prey approach seems like a really good way to get at it in a standard way. So that's super cool. That's a great caterpillar you've got there. No, I think that's super cool. I could imagine that there is apparent mutualism initially, during the emergence period, maybe May and June, as the predators are distracted, and potentially something like apparent competition later in the summer when the cicadas disappear. So if the bird populations increase numerically in response to cicada, high cicada availability during the nesting period, there might just be more predators in the system at the end of the summer. And so it seems possible you might get increased attack rates on your artificial prey later when they disappear.
Right, because there's more baby birds who are now going to be customers.
Yeah, yeah. I had this idea as a grad student for a paper titled "Top Down Till the Bottom Falls Out," where you're sort of describing this shifting predator dynamic. But yeah, I think that's a really cool idea. Like if it actually does shift between apparent mutualism and apparent competition, that would be quite cool, I think, to be able to document it. A lot of things in cicadas, just figuring out and, being organized and prepared to see it when it happens is half the battle. I think a lot of cool things happen and we're just rarely sort of positioned to observe it.
I love the way you frame sort of the way that the pulses provide insight into these trophic dynamics that are always present, right? And so the whole host parasitoid, that's just a different food web, right? That's there all the time. And then we don't really, it's hard to pick it up. It's a ton of work to document it.
It's hard to see.
And then we're having this pulse might be just enough to pull things away that will give us a glimpse into this that's happening all the time. And then to follow, we're going to follow for several years to see if the attenuation part of it that you also outlined so nicely of how long effects persist, and we don't really know, and how far do they reverberate. So it's really fun.
That's super cool. I feel like that's some of the kind of stuff that I think is most sort of intuitively interesting to me. These kind of species interaction connections. So yeah, I think that would be super cool to see. I hope your artificial prey get pecked in abundance.
John and Zoe put the first load of caterpillars out yesterday, out at Cirque, because we wanted a place, we wanted a sort of a control site where there weren't going to be cicadas as well. So
cool. Very cool.
We hope that works.
Cool. And I guess you could also, this sort of temporal dynamic, you'll probably see like a spatial dynamic as well, from sites that have a lot of cicadas to sites that have few. And that's a cool thing to make predictions about as well. It'd be cool if birds are drawn away from low cicada sites towards high cicada sites, for example. I don't know if you could see that or not, but that would be cool to look for.
Right, and the birds could just do it with their ears.
Yeah.
In an early incarnation of this study, we thought, and we'll put out fruit arrays too, and see if they don't take as much fruit. And because we've definitely heard anecdotal stories of, from my college roommate, who is about the least bug aware person that you could possibly ever imagine, but she grew up in Washington, and she said, we got to eat the cherries from our tree once in my childhood, and the birds were eating the cicadas, and so all the cherries were still there.
Wow.
I saw something from 1812 talking about the same kind of thing, that the cherries survived.
Yeah, that's very cool. That's actually very cool. I feel like I often get this question about like, are the cicadas good or bad for the forest? And I feel like the answer is often some version of it's really complicated. There's lots of pathways, and that's rarely the answer that anyone wants to hear. They'd like a simpler answer. But I think that's a really cool example. If fruit survives on the trees for longer, that would be really interesting.
Not necessarily good for the trees, right?
Not necessarily good for the trees. Yeah, absolutely. Like less seed dispersal during that year. That would be an interesting thing to try. It'd be cool to try with cherries, as a matter of fact.
We decided it was already going to take us about 40 hours per day to have worked out. So we thought maybe we would pass on until 2038.
Yeah. I mean, that's the thing with cicada work. It's very pulsed. So if you took that amount of effort and spread it out over 17 years, it wouldn't be so bad. But you do it all in the emergence year. It's a lot of work.
Mhm. I'm only going to be 80 in 2038, so it's really not a big deal.
It is something that we could deploy in August, for instance, when we're not doing most of our other surveys. We'll have a little bit more time. And that's when a lot of fruits are being produced compared to May. And then we could just compare across this year and next year and a few years in the future.
But the cicadas aren't going to be there to not eat the cherries.
Yes, but we will get the numerical response of all these extra birds all of a sudden.
Mm.
Most things aren't going to be fruiting right now anyway.
Right now, there's not the service berries should overlap a little bit. That's late May. They might be done. I don't know.
There'll be some in June. There'll be some fruits in June. Yeah. Anyway, cool to think about.
That's very cool. Well, that sounds like a great experiment, and it's cool to think about all the things that could happen.
Yeah, we might be in touch as we get things set up, and if we think, oh my God, what's going on here? Let's call Louie.
And I doubt I'll have any real answers for you. There's nothing beats sort of boots on the ground, you know, like being able to see stuff happening is pretty priceless.
The tricky part is getting the temporal part within a season and then also the between season part.
Absolutely.
That's what's so neat, is that there's these discrete temporal windows when things are shifting week to week, you know, potentially.
That's right.
And then we have this long-term thing that could be going on as well. So it's just like, I think we're just barely in time. We didn't expect it this early. We're early, a little bit early. I mean, if they're really coming out the very end of April, I mean, that's a little bit early for.
It seems early to me. I agree. I wouldn't, but I wouldn't have expected them until sort of the latter half of May. But, you know, that's based on intuition that's, you know, 17 years old at this point, and the world has changed. So
I know. So we get a climate change. That's the other question we're getting over and over again in the talks we've been giving are about what are the effects of climate change on cicadas. And we've been thinking hard about that. I'm sure you've thought hard about that as well.
Yeah.
You wrote a lot about it. So, and it just seems, you know, there's like the whole sort of late spring frost phenomenon that's going to maybe throw them off in their counting cycles, and then you know, and then there's this sort of within season they're just going to go earlier, and then this idea that Chris has said that maybe those are going to be shift to becoming 13-year because the growing season is extended. So it seems like all of those things are kind of happening simultaneously that could kind of muck things up a little bit, but but I don't know. I mean, here we are 17 years into climate change, and they look like they're going to do pretty good. So I don't know this year, so cross the fingers, so.
Yeah, I hope they come out and are numerous.
Cool. Well, thank you so much.
On that, we should probably let you get on to either childcare, or graduate students, or whatever else it is that is lurking around. But thanks so much for the seminar and for hanging around to talk. Our department loved it.
Oh, great. That's great to hear. It's been a lot of fun for me, and it's great to you know, see old friends and also meet new folks that I didn't know before. So I am totally happy to meet with anyone. You know, I guess one of the benefits of this format is it doesn't have to be, you know, today. It could be anytime. So, you know, if anyone, I'm happy to, you know.
I think Leslie would like her graduate students, Grace and Vaughn, to have a chance to talk with you. And so I was gonna, and Mariana.
Yeah,
absolutely.
I was gonna send
that would be fun.
Them a note that with the times that you had next week, or they can just write you.
Terrific. Terrific. It was a lot of fun. Thanks for the invitation to talk.
Well, thanks so much for coming.
Thank you. It was nice meeting you.
Yeah. Take care, everyone.
Bye.
Ready. Bye-bye.